So as I told you, micropropagation means, okay, producing the different plantlets in a, what do you say, defined temperature, time, as well as light. Okay, so now there are different ways by which you can do this micropropagation. So before going into that, let me just write the steps how a micropropagation is done, or the stages how it is done. So stages of micropropagation. So the first is you have to select an explant. Second, you have to, what do you say, initiate the culture and establish it. Third is, first will be the shoot multiplication. Then for that shoots, root will be developed. So root or rooting you can say. And after a shoot and root is developed in that plant, the fifth step will be what? You just have to transfer the plantlets in greenhouse. Greenhouse. Okay, so to do these things, you can use different kinds of methods. Okay, different methods. Different methods means what are the parts or how you can do this. So the first you can use is meristem. So meristem means what? Whatever the upper, what do you say, dividing part. Okay, the most actively dividing parts also you can take for this. Second is you can take a callus. Now what is a callus? When a particular explant is grown into a media, okay, it will produce a mass of cell. So this explant, when you put it into a media, it will produce what? Like this mass. So one cell is going to give what? A mass of multiple cells. So that particular thing is only called as what? Callus. So once the callus is formed, what you can do is you can transfer them into a media which is containing all the important nutrients, and you can grow the plant over there itself. Third is you can use suspensions. Okay, so suspension is nothing but liquid-like media you can use over there. So whatever the cells are there, you will put into this liquid-like media. You will provide the required air and sterile nature for them, and they will grow. Then fourth you can take is embryo. Okay, now embryo's best example I can take is if you take peanuts, means the groundnuts if you take. Okay, and that particular groundnut, if you break it from the half, here you will see a small, what do you say, a thing. Just a second. Here, if you take this, if you break it into exact half, here you will find a small thing like this. So this is the embryo of what? That particular groundnut or the peanut. So what you can do is you can take this embryo, okay, from by using a forceps and place it into that particular medium, culture medium. And again, the same procedure. Okay, you have to allow it to grow. And the fifth one is what? Protoplast. Okay, so whatever protoplast is there, by using that also, you can, what do you say, grow a new plantlet. Okay, so as compared to the other, what do you say, propagation methods, micropropagation is a well-developed method. Okay, and most probably if there is no contamination, you get good and healthy plants. Okay, so it is not necessary that you need a very large space to grow all the plantlets. Okay, plus you can put all the nutrients or you can modify the nutrients according to you. Whatever you want more, that thing you can add into it. So basically what are the advantages of the micropropagation is? Advantages. Okay, first of all, a very great or enhanced multiplication rate, wherein you can produce large quantities in short time. Okay, then whatever the shoot and root development cycle is there, or the time it is going to take to develop a shoot and root, that will also be less. Okay, so small time. That is why it's called as what? In a short time. Then because the plant is very small, okay, because they are small sized, you can easily transfer or transport them. Small size, so easy to transport and store. Okay, then whatever plants are produced is in contaminated free zone. So it will be what? Pathogen free varieties. Then it's a cost effective also, you can say. Plus you can produce what? New varieties of plant. And the very, very important thing is you don't have to depend upon any seasons. Okay, like it's let the summer come, winter, rainy. No, it's not required because you are giving the artificial conditions for it to grow. So hence it is independent of any season. So you can carry out it at any time. Okay, and the last one is healthy plants. Healthy plants. So this is all about the micro propagation. But if it is having these many advantages, some disadvantages also there. Like for example, sometimes, it's not necessary that you can take all the plants for this micro propagation. Only there are some plants you can take for the micro propagation procedure. So one disadvantage is what? You can't implement in all crops. Okay, plus there may be a acclimatizing problem. Means when you grow this plant in the controlled condition inside a test tube or any other apparatus, small vial, when you take the same plant after the growth and place it in what? Into the environment, that is your soil. So maybe there are chances that that plant could not adapt with the natural environment. Okay, the chances are very rare, but still this is a particular, disadvantage of the micro propagation. Okay, so then next we'll take is what? Cutting. So cutting, it's a basic and very common method. Okay, which most probably all the gardeners, they practice the same thing only. So in cutting, what will happen is a particular type of vegetative portion, that is, it may be a leaf, it may be a stem, root, whatever it is. Okay, you can take that a portion from the plant and you can just root it into the soil, and that will give rise to what? A new plant. So this is a basic method, that is cutting. Then next is what? Layering. So layering means here in this process there will be what? A development of adventitious root on a stem. So before this particular part is getting detached from a parent plant, that stem is induced to grow what? Adventitious root. So then what will happen is you can, with the roots, you can place that stem into the soil and it will grow into an individual part. Okay, so that is called as what? Basically the layering. And then last one is the grafting. So grafting is nothing but joining of two different plants, which is going to form one single plant. Okay, so for this you require two things. One is called as the stalk and other one is called as the scion. Now, what is the stalk and what is the scion? See, out of these two plants, one is what they are rooted in the soil. And that particular plant which you root in the soil is called as what? The stalk. Whereas another part of the plant will be having what? A shoot. So then you call it as what? A scion. So one plant will have a root, other plant will have a shoot, and together you get is what? A single plant. Okay, like for mango, roses, all this stuff, this particular grafting method is being used. Okay, so this is all about what? The types of vegetative propagation. Yeah. So second type of reproduction, that is sexual reproduction. So sexual reproduction means what? It will involve gametes, that is male and female. Okay, so basically whatever the sexual reproductions is there, like for example, how a particular zygote is formed is by this male and female gamete. It will form what? A zygote. Correct? This zygote is further going to form embryo, and then from the embryo you get an individual. So here mitosis plus meiosis will take place. Okay, so sexual reproduction means meiosis plays a very important role over here. So there are certain basic terms which you should know. Like for example, there's a term called as syngamy. Okay, so what is this term syngamy? Syngamy means there will be a complete and permanent fusion of gametes. Whereas the other term is conjugation. So conjugation is temporary union of two parents. Okay, means once that parents will exchange the male nuclei to form a synkaryon, then they will separate together and then only they will produce what? A daughter individual. So it's like both the parents come together and a bridge-like thing will be formed. That is, they will meet together. A bridge-like thing will be formed. They will exchange the nuclei and they will give rise to what? Two individual or two daughter nuclei. For example, you can take paramecium in this. Okay, then the other term is what? Parthenogenesis. Parthenogenesis. So parthenogenesis is nothing but without fertilization, without fertilization, an individual is formed. Okay, so if an egg is not fertilized by the sperm also, it will grow into an individual. So hence it's called as without fertilization reproduction. That's called as what? Parthenogenesis. Okay. So again, like you don't have this in detail, but there are again two types, that is natural and artificial parthenogenesis. Okay. And depending upon whether it's naturally occurring or artificially occurring, there are different types also according to that. Fine. So this is not there in detail for you. So we'll move on further towards the characteristics of sexual reproduction. So already you know the characteristics of asexual reproduction. So everything opposite over here will come. So sexual reproduction. Okay, first one is biparental, it should be. Okay, means two parents it should involve. Second is there should be, formation and fusion of gametes. Then both the divisions should take place, cell division. Okay. Mitotic, meiotic. So during the gamete formation, mitotic will take place, gamete formation. And during the development of the zygote, meiosis will take place. Okay, then fourth one is what? They are not genetically identical. Means they are not exact clones or copy of their parents. Then whatever the fertilization is process occurs over here, most probably will be internal fertilization. Okay, so these are what some basic features or characteristics of the sexual reproduction. Now, why is sexual reproduction very important is, as already said, for the multiplication and stability. Okay, so because there is a sexual reproduction, that is the reason evolution or heredity can be studied or maintained. Plus, you can also study what are the variations which are happening in your family, like from the ancestors till now. Okay, so if you study that particular family tree only, then only you will understand what are the changes which takes place in today's population in that particular species. So for all this purpose only, sexual reproduction is very important.